All right, hello and welcome to my tylenol-fueled pontification on castor bean detoxification. This will be castor bean detoxification starring Benjamin Bateman. So castor beans, what are they, you might be wondering? And believe me, I was wondering the same thing about 3 hours ago as well. Who needs them? I thought. Not the answer to that is not many people. They're a huge export in India, and they're a huge import in the United States and China. But they're used mainly to make castor oil, which is really good for your skin, hair, and nails. It's also a very strong laxative. It can be used as a lubricant in some industrial settings. But castor cake, which is the leftover bean gunk after the oil has been extracted, is extremely rich in proteins and other nutrients. And this would be great to feed to animals and like livestock and maybe even some people, except there's one problem: ricin. Ricin is probably one of the most accurately portrayed poisons in Hollywood. A dose of only 2 milligrams will kill you in under 24 hours, and there's 35 milligrams of ricin per 1 gram of castor bean at peak proportions. You die of horrible symptoms, diarrhea, weakness, low appetite, cramps. It's just not something you want when all you're trying to do is make your hair, skin, and nails glow. But ricin is a dimeric glycoprotein with A and B chains, which are covalently linked via a disulfide bond. The A chain is a ribosome inactivating protein, and it depurinates the first adenine in the GA sequence of the 28S ribosomal subunit. This impairs the formation of the RNA stem loop, which makes the elongation step of translation impossible. This inhibits up to 1,000 ribosomes per minute, which is why it kills you so quickly. The B chain of ricin binds to glycoproteins and glycolipids on the cellular membrane and helps make the A chain go into the cell. But wait, that's not all. There's also RCA120, which is a ricin analog, and it's also in castor beans. And it causes agglutination of mammalian red blood cells, which means the red blood cells clump together and become non-functional. It's a tetrameric protein with 2 A chains and 2 B chains, which are up to 90% similar to that of ricin. This isn't a great picture of the protein. I'm sorry about that, but it's the best I can get from the paper that I was using. So what's the solution? Well, it's just don't grow the bean. We're not growing the bean anymore. So that's it. Thanks for listening. Be sure to leave questions. I'd love to answer them. I'm only kidding. Silence the genes that are responsible for ricin synthesis in the castor beans. That's what Natalia and her colleagues sought to do in their paper, which was published in Nature Scientific Reports. So first, before we get to how they did this, I have a question for you. What do a gun, a knife, and ricin all have in common? Correct. All were tools that Walter White used to threaten his enemies in AMC's hit television show, Breaking Bad. But I have another question for you. What is different between these 3 items? I'll let you think about that for a little bit. Correct. Ricin synthesis can be inhibited with a 460 base pair intron hairpin vector. A gun cannot be. I feel like if a gun could be inhibited by a 460 base pair intron hairpin vector, that would simplify a lot of gun control. So how are we going to do this? How are they going to do this? Better question. First, you need to identify the genes involved in ricin synthesis in the first place, and there's 7 of them, by the way. Then you need to clone complementary ricin A chain sequences in both sense and missense strands of the DNA. Then this produces double-stranded RNA, which is then processed into short inhibitory RNA That's just by the cell as part of normal metabolism, and this inhibits ricin synthesis. So this is this is what they did. They treated 270 embryos with the vector, 4 took it up, and only 1 passed to the progeny. That was TB14S-5D. They confirmed this with every single technique under the sun, which is what I'm about to walk through you with. So this is a massive figure, and we're just going to walk through it A to B to C to D to E. So we'll start with A. So A is just the layout of the gene. It's a 460 base pair fragment from the ricin A chain, which was cloned in sense and missense orientation to produce double-stranded RNA under central control of 1 promoter. It also contained the reporter gene Gus, which I hope you can see where is my cursor is, and the AHAS gene, which is just for herbicide resistance. Part B are the PCR results, and as you can see, all the genes present in TB14S-5D are there, but they're not there in the wild type, this NT over here. Delta ricin is just the intron hairpin vector. Part C is expression of the Gus gene in embryonic castor beans. It's blue in TB14S-5D, which just means it's present. It's a reporter gene similar to the P-glow if you did that in Bio 202, and it changes color when exposed to a certain reagent just to tell if it's there or not. Part D is a southern blot, which identifies DNA, and the southern blot shows 2 identical copies of the gene of interest. This is good because 2 copies were cloned in sense and missense. So one of these is sense, one of these is missense. I don't think there's a way to tell which is which, but it's good that there's 2 there, and there's none in the wild type, which is what is important. Part E is a northern blot, which shows RNA. I think I misspoke. This was a southern blot. This is a northern blot. The northern blot shows RNA, and this shows that in the wild type and the TB14S-5D negative construct, there is RNA that is indicative of a ricin protein, but not in the TB14S-5D positive beans. There's no RNA indicative that ricin should be in the cell. The controls are directly to the right where my cursor is, and this is just showing that there is RNA on the membranes, just not the RNA indicative of ricin transcript. So this is all good, but how do we know whether or not ricin is actually being produced? Well, they did an ELISA. And ELISA works by putting the ricin in a well plate, treating it with an antibody linked to a reporter gene, and this is colorimetric. So the more color, the more ricin that's present. This can be quantified, and that's what the authors of the paper did. So as you can see, all of the positive TB14S-5D, all of the constructs with this hairpin vector do not produce any ricin, and the ones that are negative do, as well as the wild type control. All right, we're pretty good, right? We're done. Wait, what about RCA 120? Well, it's also been silenced. That's incredible. What? Who would have thought? Well, why? Well, the ELISA did not detect any RCA 120 being produced from the transgenic plants. Why? Well, remember, they're so similar. The proteins ricin and RCA 120 are so similar. The DNA coding for them must also be similar. So it can be predicted that the hairpin vector must also be functioning on the genes that code for RCA 120. And I know what you're thinking. You know, to really confirm this, they should probably do a hemagglutination assay. You know, that's where my mind went right away when I read this. But they're way ahead of us, so that's what they did. So as you can see, there is agglutination there's not agglutination in the transgenic plant, the TB14S-5D. So no agglutination is happening. These are all drops of blood, by the way. And the cloudy, diffuse mean there's agglutination happening of the red blood cells, and the small dot means that there's no agglutination happening, which is good, it's healthy. The PBS is just a buffer, it's a negative control, and as you can see, it's almost identical to the transgenic TB14S-5D, the construct of castor bean that should not be producing any RCA120. There's an RCA120 protein just straight purified in these wells, serving as a positive control, and there's just a wild type control here. As you go down the wells, this is a serial dilution, so the concentration of the protein gets less as you go down the wells. So now I think, if you'll agree with me, it's time to analyze some rat cells for science. So they used IEC-6, which is a rat intestine epithelial cell, and they incubated it with proteins from the transgenic and the non-transgenic castor beans. And we'll start with A. So only 15% of the transgenic cells were viable with a small amount of protein added from the non-transgenic castor beans, that is the castor beans that are producing ricin. And that is compared to 97 to 78% of the transgenic, the cells exposed to the transgenic castor beans were viable. And this was just measured by determining the viability of cells produced elsewhere in culture under identical conditions, but without any ricin being exposed to them. So then they wanted to also compare this to ribosome efficiency, because remember, ricin and RCA1, or ricin mostly affects the ribosomes. And they saw that for transgenic, for the castor beans that are not producing any ricin at all, translation is 100% efficient. But in the non-transgenic, translation is only 10% efficient at high concentrations of protein. And this was measured by using percentage of C14 leucine, the amino acid, incorporated in proteins in comparison to control cells that were not treated with protein, but left in identical conditions to grow, much in the same way as the viability percent control was measured. So now that we've analyzed rat cells, I think it's time to kill some rats for science, I guess. So Swiss Webster mice were injected with proteins from transgenic and non-transgenic castor beans. And we'll start with A. So the mice injected with the non-transgenic plants all died within 24 hours, which is really sad. But the mice injected with protein from the transgenic castor beans, that TB14S-5D, the castor beans that are not producing ricin or RCA120, didn't die at all. In fact, they became immortal and they lived forever. No, I'm only kidding. They were monitored for an extra 7 days and they developed no health complications at all, which is good. We'll move to part B. They noticed that most of the rats were dying from symptoms characteristic of hypoglycemia. So they started testing the blood glucose levels of all the rats. And they showed that the non-transgenic mice had extremely low blood sugars in hours past their injection, before they all died at hour 24. The transgenic mice exposed to the castor beans that were not producing ricin were A-OK. They had perfect blood glucose levels well within the normal range. Well, to conclude, they did it. The genes for ricin and RCA120 synthesis were effectively silenced. They did it almost too well. All the data looked excellent. Kind of suspicious in my opinion, but hey, they did it. They published in Nature. So who am I to question? Who cares? Well, now we can feed the transgenic castor cake to the animals. And using the lethal dose concentrations An animal would be able to eat 52% of its body weight in these beans that do not produce ricin or RCA 120. And I don't know about you guys, but I haven't seen anyone eat 52% of their body weight since the last time that Connor bought the Costco pack of Oreos. So animals typically eat only 2% of their body weight, especially when it comes to protein. Limit bioterrorism. Ricin is classified as a potential tool for bioterrorism by the FBI. So using transgenic beans that don't produce it could limit its use as a weapon when it's only grown commercially. And finally, farmer safety. People who just farm castor beans or harvest them can now be safer when they're working, and they can keep their jobs because their crop isn't hurting people due to ricin and RCA 120. So thanks for listening. Be sure to smash that like button and leave any questions for me in the comments. I appreciate it. God bless.